discuss the different stages of meiotic cell division. Are you ready? Let's start. Meiosis is a form of cell division by which gametes, sex cells, or germ cells with half the number of chromosomes are produced. Meiosis is also called reduction division because it undergoes two general stages, the meiosis 1 and the meiosis 2. What is fertilization? Fertilization is the fusion of egg cell and sperm cell. Take note that the egg cell and sperm cell contains haploid number of chromosomes, meaning individually they carry 23 chromosomes. When these two combine to form a zygote, they create a diploid number of chromosome, which is 23 pairs or 46 chromosomes all in all. Meiosis only occur in our sex cells, which is the sperm cell and the egg cell. These cells have half the number of chromosomes. It only occurs in our gonads, or the ovaries and the testes. Spermatogenesis is the process of sperm production, while oogenesis is the process of egg cell, ovum, or ova cell production. I have mentioned earlier that there are two general stages in meiosis, the meiosis 1 and the meiosis 2. Both of them follows the stages pro, meta, anatelo, or simply we say prophase, metaphase, anaphase, and telophase. Let's first discuss the stages in meiosis 1. Let's first discuss prophase 1. Prophase 1 is the longest and the most complex phase. It is where the chromosomes condense, synapsis occurs, where the homologous chromosomes come together to form a tetrad. A tetrad is a 2 chromosomes or 4 chromatids, sister and non-sister chromatids. So this is what synapsis looks like. The homologous chromosomes come together to form a tetrad. Take note that chromosomes play an important role in meiosis since it is considered as the physical carrier of the genetic material or the DNA. The homologous chromosomes forming a tetrad comes from your parents, specifically from your father and from your mother. These homologous chromosomes carry a specific trait, like for example, the shape of your eye, the color of your eyes, the shape of your lips, and the color of your skin, and etc. Remember that in a human being, we have 23 homologous chromosomes. From 1 to 22 are the chromosomes for our autosomes or the body cells, while the 23rd chromosome depicts or shows the germ cells or the gametes. We know that the female has an XX chromosome and a male has an XY chromosome. However, if we differentiate them, the X chromosome is bigger than the Y chromosome. Now let's go to the most important event that occurs in prophase 1, the crossing over. The crossing over are the segments of non-sister chromatids break and reattach to the other chromatid. It is where the exchange of genes occur from your parent, specifically from your mother and from your father. The site of the crossing over is what we call chiasmata or chiasma. It is where the crossing over occurs. Now this is how the crossing over occurs. The crossing over provides a variation in terms of human as well as in animals or even in plants. Now let us summarize the events in the prophase 1. Chromosomes become visible, nuclear envelope breaks down, synapses tetrads form, and crossing over occurs in the chiasmata. Now let's proceed to metaphase 1. It is the shortest phase in meiosis. Tetrads align on the metaphase plate. It looks like this. Also, in metaphase 1, pairs of homologous chromosomes move to the center or to the equator of the cell to form a metaphase plate. Next is the anaphase 1, Homologous chromosomes separate and move toward the poles. The sister chromatids remain attached at their centromere. Homologous chromosomes move to the opposite sides of the cell, or in the process called disjunction. So the cell looks like this. The last stage of meiosis 1, the telophase 1. Each pole now has a haploid set of chromosomes. Cytokinesis and two haploid daughter cells are formed. Take note that cytokinesis is the process where the cytoplasm divides. Chromosomes gather at the poles of the cell, cytoplasm divides, creating two cells. Now these are the product of telophase 1. Each cell contains an haploid number of chromosomes, or 23 chromosomes. We are done with meiosis 1. Let us now proceed to meiosis 2. In meiosis 2, we still follow the 4 stages: the prophase 2, the metaphase 2, the anaphase 2, and the telophase 2. Let's discuss them one by one. In prophase 2, it is seen as prophase in mitosis. A new spindle fiber forms around the chromosomes, and the nuclear envelope breaks down. The prophase 2 looks like this. In metaphase 2, it is also the same with the mitosis. Chromosomes line up at the equator, forming a metaphase plate. In anaphase 2, it is also the same with anaphase in mitosis. The sister chromatids separate, centromere divides, and the sister chromatids move in opposite poles of the cell. It looks like this. In telophase 2, it is also the same with telophase in mitosis. The nuclei form, cytokinesis occurs, and nuclear envelope forms around each set of chromosomes. Spindle disappears, cytoplasm divides, creating 4 haploid daughter cells. So in all, the products of meiosis are 4 haploid daughter cells. Let us summarize what we know by looking at this figure. From diploid cell, the cell undergo the first stage of meiosis, which is the meiosis 1. And the resulting products are 2 haploid daughter cells. From 2 haploid daughter cells, it undergoes the second meiosis, with the result of 4 haploid daughter cells, meaning the products of meiosis are 4 haploid daughter cells. Just take note that meiosis is called reduction division, from diploid cell to haploid cell, as well as from diploid number of chromosomes to haploid number of chromosomes. So yes, we're done with the stages of meiosis, the meiosis 1 and the meiosis 2. I hope you learned something from this video. If you have any questions, please send me a personal message in Schoology. Thank you everyone.